In this video, we're going to discuss three main topics: phylogenetic trees, speciation, and the concept of fossil genes, sometimes called pseudogenes. This is a very simple and abstract version of a phylogenetic tree. And its main purpose is to demonstrate the evolutionary relationships between extant species. The term extant means that they are species that are currently alive today, and that's opposite to extinct species that no longer exist. And when we talk about evolutionary relationships, what we're talking about is how recent or how distant in time do two extant species share a common ancestor. So, for example, the species that is represented by this shape and this species that's represented by this shape share a common ancestor right here. And that's more recent in time. So time is the length of these bars here. That's more recent in evolutionary time than these two share a common ancestor with this orange guy here, which is way back here. So what we say then is these two are more evolutionarily related to each other than they are to this guy here in orange. Now, the question is, how do we determine this? What is it about each of these three species that allows us to determine which ones are more evolutionarily related to each other and which ones are less? And the main way we do that nowadays is by sequencing the DNA, the genomes of the various species, and comparing that genomic sequence to one another. So again, what I have here is a very abstracted version of that. So instead of having the sequence of DNA, A's and G's and T's and C's, I've just abstracted that out to the alphabet here. And so when we sequence a specific region of DNA, so sometimes, for example, mitochondrial DNA, and that's what you're going to be doing in lab, what we might see is this. Here's that sequence within the purple species, here's the sequence here in the blue, and then in the orange. And you'll notice that the purple and the blue share A, B, C, D, and E. A, B, C, D, and E. That's much more in common than with orange, which only has A, B, C in common. So we know that the purple and the blue are have more recently evolved from a common ancestor than that with the orange. So we have the common ancestor more recent in time down here. However, we can see that the purple and the blue are evolutionarily related to that in orange. It's just more distant. Right? All three of them have ABC, ABC, ABC. So we have then another common ancestor up here, farther away in time, right? More distant in time, that is in common to all three of these extant species. And even though the species represented by this node here and the species represented by this node here are extinct, based on these extant species that have evolved from them, we can start to make some guesses, some hypotheses as to what their DNA sequence was. For example, we know this species here must have the A, the B, the C, the D, and the E sequence. We don't know whether it had an F or whether it had a G. We would have to maybe look at some other extant species to figure that out. And we know that this species here must have had an A, a B, a C. Now, we don't know necessarily what else it might have had. Again, we'd have to look at a greater number of different extant species to get a better idea of what the rest might be. But for now, we at least know this. So, because all three species evolved from this common ancestor here, they all have A, B, C. And because these two guys, the purple and the blue, evolved from this here, we know that they must have A, B, C, D, E. And so these are the sorts of things that you can analyze and understand using phylogenetic trees. Now, let's talk about speciation. How did we get the purple, the blue, and the orange species here? How does speciation work? How are new species made? Well, we start with a common ancestor. So here it is right here. Now, of course, this is only part of a complete organismal life phylogenetic tree, just a very, very small part. And so there's lots of other common ancestors up here, but let's just start with the one that's common to these three here. And this node here represents a population, a particular species, a single species. And the node itself represents a very important time in evolution history, a time at which that species, that community, that population gets separated, such that, okay, part of that species, part of that population is over here, part of that population is over here, and they're separated by all sorts of possibilities. They might be separated by location, where maybe a mountain arises and part of the population becomes separated from another population. But there's all sorts of other ways in which the two communities, one population is broken into two. But the key point here is that any mutation that arises in one part of this population, it does not necessarily mix and occur within the other, right? They are separated. They don't mate with one another anymore. They're still the same species. They've just become separated from one another. Okay, so then what happens here is, okay, this population here then goes through time. And we know that as time goes on, as generation after generation occurs, mutations occur. Mutations are changes in the sequence of DNA, which causes changes in the amino acid, or can cause changes in the amino acid sequence of the proteins, which can cause the structure of those proteins to change, which can cause the function of those proteins to change, which can cause new traits to emerge within the population. Now, those two new traits may either be selected for, which means that they will continue to spread within the population, or they will be selected against, meaning they will dwindle and be purged from the population. Now, what traits are selected for and what traits are selected against? That is dependent upon the current environment in which the population exists. So, as time goes on, that population is going to be developing new mutations, causing new traits, some selected for, some selected against, and will be adapting to their current environment. And so let's go here. So by the time we look all the way down here, these guys here, this population no longer necessarily looks like it did up here in this node here. We've had all of this time to adapt to the environment in various ways by having mutations occur, which cause new traits, which are selected for and selected against. Here, this population now, same thing, might not look the same as it did here because we've had some time for adaptation to occur. We've had lots of time where mutations can occur, leading to new sequences of DNA, new amino acid sequences and proteins, new structure, new function, new traits, selection for, selecting against, and so this population might not look like that population here. And then we have another speciation event. We have another point in time where this population is separated into two, where they no longer mate with one another. And now any mutations that occur here leading to our purple will not necessarily spread into the community here leading to the blue. And so new mutations occur, new traits occur, selected for, selected against, and we have then two new species. That, in a nutshell, is how speciation works. So let's look at an interesting example. And this is also an example that is going to demonstrate an interesting concept called fossil genes or pseudogenes, or the term fossilization. Let's take a look at some species of fish. And they are all relatively related evolutionarily to one another by this common ancestor here. And when we go and sequence the hemoglobin gene, so the gene that encodes a protein called hemoglobin, that protein is necessary in red blood cells in order to carry and transport oxygen. When we sequence the gene that encodes hemoglobin in these three fish species, what we see is we've got hemoglobin here, okay? We've got hemoglobin here, and so I've just spelled it a little bit differently, just to basically, it's an abstracted way to say the structure of the protein is the same, basically, just a little bit different, a little small amino acid change due to a mutation, but it still has the same function, which is it can carry oxygen. But then you look here, and this fish here, when you sequence the hemoglobin gene, it's very different. And in fact, it has deletions within the gene. It has very different amino acid sequences. And what it turns out is the hemoglobin gene is not even expressed anymore. It doesn't even make the protein. It's mutated in such a way, it has degraded in such a way where it does not even encode protein. We can still detect that, yes, indeed, there used to be a hemoglobin gene. We can see the E, the M, the O here, but there's a lot of other mutations. And what we call a gene that no longer functions, that has accumulated so many mutations, we call it a fossil gene or a pseudogene. Okay, so here hemoglobin, normal gene, encodes a protein, fantastic. Same thing here. Here, it's become fossilized. So let's take a look and see why, right? What is it about the environment in which this population of fish found itself that would select for, or would allow for, I should say, allow for the fossilization of the hemoglobin gene? So the idea, we know, since this species here has a functional hemoglobin gene, since this species here also has a functional hemoglobin gene, then we know that this common ancestor between all three must have also had a functional hemoglobin gene, right? Maybe, I guess you could argue that maybe this common ancestor had a dysfunctional or a fossilized hemoglobin gene, and that this one independently evolved a functional hemoglobin gene, and this one as well independently evolved a functional hemoglobin gene. But that has to have occurred on two separate occasions, so that's very unlikely. And so what we do is we look at what's the most likely event. And the most likely event is this common ancestor had a normal hemoglobin gene, right, that was retained here in this species, was retained here in this species, and was lost here in this species. So let's look at the evolution of this species here. What happened to allow for the fossilization of the hemoglobin gene? Well, let's take a look. Well, what we know is back in evolutionary time, so millions of years ago, this population of fish separated, got separated, where one part of the population remained in relatively warm waters, and the other population got separated into waters that became very, very cold. And so now we have two different environments, and that shift in environment is we're going to be able to detect it in the sequence of DNA of the genomes of these two fish. What do I mean by that? Well, what we know happened is, all right, in that shift to cold water, the blood of these fish got very viscous, very thick, right? The colder liquid is, the thicker it's going to be. Well, a mutation occurred. Okay? And remember, mutations are random. This is just totally by chance. A mutation occurred that eliminated the ability of these fish to make red blood cells. And that allows the blood to be less thick, right? So now it can still travel through the veins. And now oxygen isn't transported via the red blood cells. It's transported via just being dissolved within the plasma of the blood. And that is enough to allow those fish to have enough oxygen to survive. Remember, red blood cells contain hemoglobin. This is a protein that allows the red blood cells to carry oxygen, right? And hemoglobin is encoded by the hemoglobin gene. So if the fish no longer make red blood cells, they no longer need the hemoglobin gene. Which means at this point on, right, if a mutation occurred within the hemoglobin gene, normally if the fish required that hemoglobin, right? So let's say a mutation here occurs within a hemoglobin gene. Well, that fish is in the warm water, still has red blood cells, still really requires hemoglobin. Any mutation that knocks out the ability of hemoglobin to function properly will be selected against and purged from the population. Here, because the fish don't have red blood cells, they don't need hemoglobin. When a mutation occurs in the hemoglobin, that mutation is not selected against, and it stays within the population. All right, generation. Another mutation occurs within hemoglobin. Maybe a big deletion, like a big mutation, like a deletion. Well, same thing. Because the fish no longer make red blood cells, they don't need hemoglobin, and that mutation is not selected against and therefore spreads within the population. And year after year, generation after generation, mutations are occurring within the hemoglobin gene and are not being purged, are not being selected against because the fish don't need hemoglobin. So by the end of it, right, if we look at ice fish today, what we see is this degraded hemoglobin gene that no longer is even transcribed and translated into protein. Now, all of these mutations that are occurring, this is really, really, really important to know. All of the mutations that are occurring in that hemoglobin gene in this time period here, there's mutations just like that are occurring here, right? Mutations are random. They're just constantly happening. They're happening all over and there's all within the genome. There's no conscious thought as to where they're occurring. They just are. So mutations, even in the population leading to this extant species here, plenty of mutations occurring to hemoglobin, but That population exists in an environment in which hemoglobin is absolutely essential. So any mutation that occurs that breaks hemoglobin is selected against, and those individuals die and are purged from the population. Thus, by the end of it, all we have left are the individuals that still retain a functional hemoglobin gene. It goes the same for these fish here. It's only these fish that have shifted in a new environment that allows them to fossilize their hemoglobin gene. Okay. So what we say is that in this particular fish here, the hemoglobin gene has become fossilized. It is now a pseudo gene. And you can make a prediction. We can say, hey, if we took this species of fish and we look 10,000 years from now, what would we predict if we tried to sequence the hemoglobin gene? What we would probably predict, what we would probably see, is we would no longer even be able to detect that there was ever a hemoglobin gene there in the first place. That it would have accumulated so many mutations that we wouldn't even see chunks of it that has any similarity to the hemoglobin gene here or the hemoglobin gene there. Okay, so hopefully this will help you in understanding your assignment today, because you're going to be looking at basically the same idea, the fossilization of a particular gene and comparing that gene to evolutionarily related organisms and making some predictions based on that.